We'll start by discussing cytarabine arabinoside. To understand its function, we need to recall some fundamental biology. Recall how DNA differs from RNA. Yes, the nucleotide base is different in thymine versus uracil, but the sugar portion is more important here, in that RNA is ribose, where deoxyribose is part of the DNA. The difference being between the two that the second carbon has lost an oxygen with deoxyribose. So now you know where the deoxyribo portion comes from in the term DNA. The nucleotide itself is comprised of a phosphate group, the deoxyribose group, and then the base, which is either a purine or a pyrimidine. The pyrimidine base that I want to focus on is cytosine. If you look at the structure of cytosine compared to cytarabine, you can see that the structure is largely similar, except that the second carbon on cytarabine is now oxygenated. Because of the added oxygen, the ribose sugar of cytosine now becomes chiral, making it an arabinose sugar. So the name cytarabine arabinoside comes from the makeup of cytarabine for the base and arabinose for the sugar. The last thing we will talk about is the cell cycle. Recall that the S phase is where DNA synthesis occurs. So after mitosis and during the G1 phase, the chromosome is not double-stranded until the S phase, where it replicates. It is during this process that cytarabine will be inserted into the DNA, ultimately leading to cell death, as the arabinose sugar, due to its chiral shape, does not allow for proper rotation of the DNA. This is why cytarabine is termed an antimetabolite and S phase specific. Next, we will discuss the mechanism of action for cyclosporine. Before discussing how cyclosporine works, we will first discuss some of the physiology of T cells. Here's a cartoon representation of an antigen-presenting cell binding to a T cell receptor. Once the antigen is bound, this results in a cascade of reactions that end in the release of intracellular calcium ions. The release of calcium ions binds to calmodulin and the beta subunit of calcineurin enzymes. The binding of the beta subunit allows for a conformational change that allows the calcium calmodulin complex to bind to the inhibitory domain of calcineurin. This then allows the catalytic domain to function on calcineurin, and it will dephosphorylate target proteins. In this case, nuclear factor of activated thrombocytes, or NFAT. Once dephosphorylated, NFAT becomes activated, revealing the nuclear localization sequence, or NLS. NFAT then translocates to the nucleus and attaches to specific enhancer regions on DNA, triggering transcriptions of particular genes, where in T lymphocytes, NFAT regulates expression of interleukin-2 cytokines. An endogenous protein that we have not discussed yet is cyclophilin. This is where cyclosporine will actually bind to. The drug cyclophilin dimer then binds extremely to calcineurin, inhibiting the binding of calmodulin and thus the dephosphorylation of NFAT, where it remains cytosolic. Therefore, interleukin-2 is not produced by the T cells.